Hello everyone, I Anish. I welcome you all on the behalf of A.S.S Foundation. As you all know, we are starting our new batch from July 11. So those who have not registered till now, please do it ASAP. Today we will be discussing about the antigens and immunogens and the antigenicity and the immunogenicity. Next is the types of antigens, what are heptanes, and the factors that affect immunogenicity, adjuvants, and what are epitopes and paratopes. Let us begin with antigens and immunogens. So, antigens are the particular molecules which can be specifically recognized and bound by an antibody, and their property is known as antigenicity. Okay, antigens are the ligands that reacts with the products of the immune responses to, and the term antigenicity is the ability to induce a immune response and the ability to react with the products of that response. The next is immunogen. Immunogen is a molecule which can elicit the production of specific antibody upon injection into a suitable host, and the property is known as immunogenicity. That is the inherent ability of substance to induce a specific immune response. All immunogens are antigens, but not all antigens can be immunogens. This is an important statement and which is asked in many exams. All the molecules are immunogenic and are also antigenic, but not the opposite is correct. For example, heptanes. Heptanes are antigenic but not immunogenic. Now next we will be discussing the types of antigens. This classification is based on the source, like where the antigen is coming from. The first one is xenoantigen. It is the type of antigen that is found more than in one species. Next one is alloantigen. These antigens are present in allelic forms encoded at same genus in different individuals, like ABO blood grouping in humans. Next one is autoantigen. Autoantigen are the causes of autoimmunity when they are present in the same individual as tissues or organs, but the incapability to identify or recognize themselves as the self cells, then these type of antigens act as autoantigens. The next one is heterophile antigen. These are the similar antigens present on dissimilar organisms. See, now we will be discussing about the heptane molecules. Heptanes are that particular molecules when they are covalently bound to a carrier protein, they can induce the immune responses. But heptanes itself cannot induce particularly the immune responses. That is why heptanes can be antigenic, but they are not immunogenic. So, heptanes are the molecules which themselves do not induce immune responses, but when they are covalently bound to a large carrier protein, they can induce an immune response. Here in the figure, a heptane molecule is conjugated with a carrier protein, and depending upon the which type of carrier protein, the antibodies are formed. Like when only heptane is induced, there are no antibody formation. When only protein, then there are anti-BSA antibodies. And when heptane is conjugated with carrier protein, like heptane is DNP and the protein is BSA, then there are anti-DNP, anti-BSA antibodies formation. So next we are discussing the factors that affect the immunogenicity. The first one is foreignness, the next is molecular weight of an immunogen, next is chemical composition, and the last one is susceptibility to antigen processing. So let us discuss all these factors one by one. foreignness, lymphocytes that do not bind to self antigens are allowed to further develop, therefore, they will later only recognize non self antigens. An antigen must be foreign or alien to the host which it makes contact. The greater the phylogenetic differences, the more the foreign something becomes, and the greater is the immunogenicity. For example, BSA is not immunogenic when injected into cow because it is extracted from cattle itself. But when it is injected into chicken, then it is immunogenic. The next one is molecular size. There are good and poor immunogens depending upon the molecular size. The immunogens who have 5 kd to 10 kd molecular weight, they are poor immunogens. For active or good immunogens, the molecular weight should be around 100,000 kilodalton. Okay. Now the next one is chemical composition. The chemical complexity of a molecule contributes significantly to its immunogenicity. We will be discussing all the biomolecules complexity to a immunogen. For example, in proteins, there are large array of building blocks like only 20 amino acids. This diversity imparts epitopes of differing specificity to a molecule. Immune responses usually are induced only after prolonged exposure to antigens or by the use of adjuvants. Adjuvants are the molecules that enhances the immune responses. The next one is polysaccharides. Polysaccharides are almost non-immunogenic. They can impart a unique specificity to antigen if appropriately coupled to a carrier molecule. That is, they require a conjugate protein to become an immunogen. The next one is nucleic acids. Nucleic acids are also considered to be non-immunogenic, but when they are combined with basic proteins, they act as immunogens. Okay, and lipids are heptanes and needed to be conjugated with carrier proteins to produce antibodies. Now the next one is susceptibility to antigen processing. Large insoluble macromolecules are more likely to be phagocytized for the processing, so there are more chances of them to be immunogenic. Now the next one is adjuvants. Adjuvants enhance the immunogenicity. They are recognized by the toll-like receptor TLRs, and the example of it is aluminum hydroxides, which are given with the toxins of DPT vaccines to enhance the immune response. And last, we will be discussing about the epitope and the paratope. Epitopes are the sites either or within the antigen with which antibodies and T cell receptors react. Okay, as it is seen in the figure. And while paratope, these are antibodies. Antibodies are specific for epitopes, and the area of antibody molecule that interact with epitope is called the paratope. Epitope is present on the antigen while paratope is present on the antibody. And these epitopes are immunoreactive only if they are spatially accessible as a result of tertiary protein structure. So there are some terms that are related with epitopes. Epitopes needed like the valence of an antigens. Antigens are multivalent structures, and the size epitopes are very small. They are just 4 or 5 amino acids or monosaccharide residues. And when we talk about the site, some antibody binding sites, that is epitopes, are on the antigen surfaces, whereas others are internal. Internal epitopes are expressed only after the antigen has been partially degraded Okay, that's all for today. In the next lecture, we will be discussing about antibodies.